Hello students, this is Molecular Basis of Inheritance Part 9. So in this slide we are going to talk about the experimental confirmation of semi-conservative type of replication of DNA. Okay, and it was proved by these two scientists, that is Meselson and Stahl. These two scientists, they proved that when the DNA it gets replicated, that means when the old DNA it forms two new strands of itself, exact copies of itself, then out of these two strands of newly formed DNA, one strand of the DNA is old strand, that is called as parental strand, while the other strand is called as daughter strand, or that strand is also known as newly formed strand. Okay, so the old strand is the template strand. So in the semi-conservative replication, at the end of the replication, two DNA daughter molecules they get formed, and these two newly formed DNAs, they are exact replicas or exact copies of the old DNA strand. In each DNA daughter DNA, one strand is parental, that is the old one, while the other is totally newly synthesized strand. So that's why as the half or 50% of the DNA is old strand, while half portion of the DNA, that is 50% of the DNA, is newly formed, and because of that this process is called as semi-conservative replication, and that was experimentally proved by these two scientists, that is Meselson and Stahl, and their experiment is known as Meselson and Stahl experiment. So in this slide we are going to discuss about what is a density gradient centrifugation and what are the isotopes. What is the difference between that? You can observe in this diagram the interpretation of results of Meselson and Stahl experiment on the separation of DNA by equilibrium density gradient centrifugation. So the Meselson and Stahl, they used this density gradient centrifugation method. So in the Meselson and Stahl experiment, it is the experiment which supported the Watson and Crick hypothesis that DNA replication was semi-conservative type of replication. In the semi-conservative replication, when the DNA double-stranded DNA helix is replicated, or when it gets copied, each of the two new double-stranded helices, out of these two helices, you can observe in this diagram, one is newly synthesized, one is red in color, and one is green in color. So that red in color is the old DNA and the newly formed DNA is green in color. Okay, so this experiment, it proves that by using the density gradient centrifugation, the Meselson and Stahl, they proved that this type of replication, DNA replication, is a semi-conservative type of replication, and it has been called the most beautiful experiment in the biology. So Meselson and Stahl, they decided the best way to tag the parent DNA would be to change one of the atoms in the parent DNA molecule. So what they used, the technique that they tagged the atom, one of the atom of the parental DNA. So they used two types of tags. They used two types of isotopes, N14 and N15, as nitrogen is present in abundant amount in the DNA structure. If you have watched the very first lecture of this molecular basis of inheritance, so I have explained in that video that what are the nitrogen bases, the structure of the DNA. In that you will come to know that the nitrogen is present in large amount in the DNA bases. Okay, so since the nitrogen is found in the nitrogen bases of each of the nucleotides, so they decided to use an isotope of the nitrogen to distinguish between the parent and the newly copied DNA, that is the old strand and the newly synthesized DNA. The isotope of the nitrogen had an extra neutron in the nucleus, which made it heavier. So the nitrogen N15 is heavier than the N14. N14 is comparatively lighter in weight than the N15. So what is density gradient centrifugation? So I have explained the centrifugation method in my earlier lecture. I have explained what is the importance of centrifugation, how it is carried out in the lab, and with the help of which machines it is carried out in the lab. So you can check out the earlier lectures. So what is density gradient centrifugation? A method where the components of a sample are separated on the basis of their density in a dense medium or density gradient in a centrifuge according to the centrifugal force. So in the density gradient centrifugation, ultracentrifugation technique, where the centrifugal force gets applied to this centrifugal tube. This centrifugal tube contains the sample, and according to the molecular weight or according to the mass, according to the density, the molecules or the components which are present in this medium, they get separated. So you can observe the difference between this first, second and the third test tube. Now observe this first DNA strand, it contains only N15 type of DNA. That is the DNA which is shown in this diagram. This DNA contains only N15 type of isotope. As N15 is heavier, so you can observe the band. It is towards the bottom of the test tube. Okay. So this is heavy. Uh, the same if you observe in the generation 1, this we are going to discuss in the next part, that this bacterial cell, these bacterial cells, some of the sample, it is transferred to the N14 medium. So after 20 minutes, that bacterial cells, that culture is called as generation 1 or first generation. So when the sample was taken from this culture, that is from the generation 1, it was observed that it contains hybrid type of DNA. So hybrid means out of these new replicated DNA, one strand is newly synthesized, another is old strand. So old strand contains N15 type of isotope, as this old strand comes from this parental DNA. And the newly green colored strand is the daughter strand or newly synthesized, which gets formed by using this N14 type of isotope. So this is the band which they observed by using this density gradient centrifugation when the centrifugal force was applied. So as you can observe, there is change in the positioning of the band too. It is to the lowest part of the centrifugal tube, right? While this is present, it is somewhere middle of the test tube. So it as why the position is getting changed, because the molecular weight is different. The density of the sample or the components which were present in the centrifugal force, that their density is different. So they are hybrid. N14 and N15, both the isotopes, they are present in the medium. After this, when the after 40 minutes, the second generation was tested. So the results were completely different. So they were they observed the two types of bands, one with the light band and other with the hybrid. So hybrid band contains the N14 and N15, both the isotopes, while the light stranded because of the newly formed uh, DNA. Okay, so you can observe here in the second generation we are getting a completely new, that is N14, containing both the strands of the DNA, they are N14, and here both they are hybrid, while in this again they are N14. So if you, why this type of result, if you observe this diagram carefully, see this is the first generation. When this you observe this first diagram, both the strands are red in color, both of them they contain N15 type of isotope. When this culture was transferred to the medium which was containing N14 type of DNA, in this part what happened, one strand was with the parent strand, that is that culture was with hybrid DNA, as one with the N15 and the newly synthesized DNA was with the N14. Okay, that much we can understand. Now after 40 minutes, when the second generation you observe, why both the strands are green in color, try to understand this. When now this red and green, if they get separated, now this green strand is the old strand. This green strand, here is the presence of green strand, and another is the red strand. Now this red strand is absent here because here newly synthesized strand is again a green strand in this diagram. Again, if you are getting this or not, see, try to understand this. If you take this one strand only, okay, so in this, out of these two strands, observe this, out of these two strands, if you take only one strand of this double stranded DNA, so this one strand is with the this part, green part, and one strand is the red strand. So this red strand goes here, green strand and red strand. Again, out of these red and green, red strand is here and this green strand is here. Okay, so there is separation of red and green. So as the newly synthesized DNA you are talking about in the second generation, now in this, as the green strand is, this green strand is the old green strand and the newly synthesized is again a green strand. In the second strand, the red strand is the old parental strand and the green is the newly synthesized. Again, in the third diagram, if you are observing in the third diagram, the red is with the old strand, okay, and the another is with the green strand, and 14. Again, this green strand with the parental strand and the newly formed is with the daughter strand. Okay, so here you are talking about in the second generation, there is completely mixing of the DNA strand. Out of all these four strands, if you are observing all these 1, 2, 3, 4 diagrams, you will observe the difference. This green colored strand is containing only N14 DNA. These two are containing N14, N15 type of isotopes of the DNA. These two are hybrids, while the third one is with the N14 DNA. So there are chances of getting only two types of bands. Okay, let's try to understand this much better in the next slide. So in this slide, we will try to understand what was the Meselson and Stahl hypothesis. So there were three postulates, three theories, three methods of DNA replication which were proposed at the time of Meselson and Stahl. First is the semi-conservative replication that we have discussed here. Right now we are discussing it, that in the semi-conservative type of method, this is double-stranded DNA. Out of these two strands, when this DNA get replicated, then one strand is the old strand. You can observe here, one is with a different color, another is the newly formed DNA. So 50% is the newly formed DNA and 50% is the old strand. Okay. Another second postulate was the conservative type. So conservative type or method of DNA replication here, it was postulated that, now see, if in the conservative, if you are talking about this is the double stranded DNA. So here after replication, it was supposed or it was proposed by a few scientists that two DNA, when they are getting formed by the DNA replication process, one is completely newly formed DNA and other is completely old strand. So this is completely new strand and this is this is completely old strand, while this is completely new. So 100% old strand and 100% new strand gets formed, which was again proved false. The dispersive type of postulate, now this is the third dispersive type of method of DNA replication was proposed. So in this type of postulate, you can observe in this diagram, this is the old DNA strand, and in the dispersive mode, when the DNA gets replicated, it was postulated or it was put forth by some scientists that some part of the DNA, it is old one, some is newly formed, and again some part is the old strand. You can observe here the newly formed DNA, it is the old strand, it is the new strand. So in the dispersive mode of replication, it was proved by some, it was suggested by some scientists that there is mixture of the DNA strand. Some part is, some segment of the newly formed DNA is, some part is old, some part is new. So it is present in the form of segments, it is scattered one. Okay, so this conservative and dispersive, these two methods, they were these are not biologically that much important. So these two postulates, they were not used by or they were not used by the scientists, and this dispersive as well as conservative. So Meselson and Stahl, they proved that the correct method of DNA replication is the semi-conservative replication. Here you talk about one newly formed strand and one is the old strand. So what was the Meselson and Stahl hypothesis now? They cultured bacteria. They used Escherichia coli bacteria, that is E. coli bacterial cells in the N15 medium. So first they cultured the bacterial cells in containing the N15 medium. So whatever bacterial cells they were grown, they were allowed to grow in the N15 medium. So all the bacterial cells, the DNA of those bacterial cells will contain definitely the N15 type of isotope. Okay, so that we have discussed in the earlier slide. So they will synthesize N15 is a heavy isotope of nitrogen, so the DNA is of heavy density. So that we have observed in the first test tube. The band, the density gradient centrifugation band was completely lower part of the centrifugal tube. Then they shifted the bacterial cell to the N14 medium. So again, the first generation you have observed in the previous slide. So in the first generation we have seen that the density gradient centrifugation, the N14 N15 hybrid type of band, it was observed. So DNA was isolated at different times corresponding to the replication cycles. So at a different particular type of time break, they observed or they isolated the bacterial cells, and they observed it in the zero cycle, zero time period, one generation, first generation and the second generation. That's all. And that they proved that the DNA replication is semi-conservative type of replication. In this slide, we will try to understand that why did Meselson and Stahl they use density uh, gradient method, or why they use the density label in their experiment, why they labeled the N14 and N15. So to describe how DNA replicated, Meselson and Stahl needed to distinguish between the parental and daughter DNA molecule. So to separate the parental DNA molecule from the daughter DNA molecule, it was very much needed by the Meselson and Stahl to distinguish between these two strands. Now you can observe, now this is the E. coli cell which contains N15 type of isotope, and this is another kind of E. coli cell which was again cultured in different broad culture, right? These are the broad cultures. So it contains N14 type of medium. So if you observe it carefully, the E. coli cells which were collected from this medium, which was containing the N15 type of isotope, all the bacterial cells of the E. coli, they contain only this N15 type of DNA in their cell. Okay, and if you observe the another kind of E. coli cells which are grown in the N14 type of broad culture or in the N14 medium, all the bacterial cells, they will contain only N14 type of isotope in their DNA. So that is the basic difference. That was the basic difference that the Meselson and Stahl they wanted to prove, because it was very much needed for them to prove that how the N15 and N14, as their masses are different, So after using the centrifugal force, they will form different kinds of bands. So you can observe this is the ultra centrifuge tube. Now this is cesium chloride medium in which the density gradient method was used by them. So here the different types of bands they were observed by them. So on the basis of this observable result, as you can observe in this diagram, okay, this is the density gradient technique that proved that they used different kinds of N15 and N14 isotopes and they formed different kinds of bands. They achieved that by modifying the molecules, so each kind had different density. So this N15, when this culture was ultra centrifuge, as N15 is heavier, so it will form the band at the base of the centrifuge, and as the N15 is heavier in weight. If the bacterial cells, if they are containing both N15 and N14 type of uh, isotopes in them, then they will form the band at this position as they are hybrid. What do you mean by hybrid? Hybrid means that bacterial cells, they contain both types of N15 as well as N14 types of isotopes. If you are talking about only N14 type of isotope, as the N14 type of isotope is comparatively, it is lighter than N15, so it will form the band at different positions. So that was proved experimentally by this Meselson and Stahl, that by using the density gradient method, by using the density leveling N15 and N14, they were able to prove that the DNA gets replicated by semi-conservative type of replication. So this is the summary of Meselson and Stahl experiment. See, what is the hypothesis? DNA replicates semi-conservatively. That was their hypothesis. That after the replication of DNA, one strand is old one, while the other strand is newly formed. So what method they used? This is one flask, this is another flask. This blue colored flask is it contains N15 type of medium, that is it contains heavy isotope. N15 is heavy isotope. So they allowed the growth of bacterial cells in this medium. Then they took this sample in the 0 minute when the bacterial cells, they were grown in this medium. So they ultra centrifuged this medium. By this density gradient method, what they observed, that all the DNA in the bacterial cells, the DNA was heavy. It contains only N15 type of isotope. So the band, you can observe this band is at the base of the centrifugal tube. So you can observe this is the newly formed DNA strand, which is again blue in color. So both the strands, they contain N15 type of the isotope. When after some time, after 20 minutes, they transferred the same E. coli cell into the next broth or in the next medium. So they transferred some bacterial cells to this N14. Now this red colored medium is containing N14 type of isotope. Now they collected some sample from this after 20 minutes, and they allowed the density gradient technique or density gradient centrifuge to this tube. In the first generation, all the bacterial cells, they contain all intermediate type of DNA. That means all the bacterial cells, they were having the parental DNA with N15, while the newly strand or newly formed DNA was with the N14. In the next generation, that is in the second generation, when the sample was collected after 40 minutes. Why? Because 20 minutes after the one round of replication, this is the first generation. After 40 minutes, this is the second generation. What they observed, they got two different kinds of bands in the centrifugal tube, one with the intermediate DNA and another with the lighter DNA. So that means some of the bacterial cells, they were having the intermediate, that is one with the N14 and one with the N15 type of isotope of the DNA, while some of the bacterial cells, they were carrying only N14 type of the bacterial cells. So that means they were having the lighter DNA. So the pattern could only have been observed. So this type of pattern is completely observable in the observable form. So the hypothesis which was put forth that DNA gets replicated by semi-conservative type of replication mode, it was proved by the Meselson and Stahl experiment. So that's all for today's session. Thank you.